Hi. It's just such a pleasure to be part of this conference, and I want to thank Aviva and Cindy to organize this conference. I think there are some momentous things happening at this conference, and it is the level of engagement I haven't seen for almost a couple of decades. So thank you for all you do. And I think it's such a fitting tribute, if you like, to Dr. John Barringer, who passed away about 10 years ago, having made sustained contributions to Gaucher disease for half a century. And really, the contributions he made have been transformative. And in a sense, we stand on the shoulders of giants like him and Dr. Roscoe Brady. So I'm going to be talking about the brain in Gaucher disease. And all of you know, all of you here, I think you will accept that people who are affected with Gaucher disease are really quite gifted individuals in their diverse ways. Even patients with type 3 Gaucher disease, I have patients who are barely 14 years old, performing at college level math and so on. CEOs of companies, 40-year-olds. So the topic of how GBA mutations affect the brain is very important. But I want to say that one of the most profound privilege of my career in life has been part of the Gaucher community. It started in England, and then I've had the opportunity to work with the Gaucher community across several continents. And everything in our program starts with a patient. And all big scientific questions that I have addressed in my career have begun with an interaction with the patient who has asked me the questions. And so the Gaucher community are integral partners in the discovery that we subsequently make. So we also want to develop a concept of lifetime of care, how Gaucher disease expresses itself through the lifespan, because it has very protean manifestations depending on if you're at the older age and there are problems related to that, whereas teenage, we heard some of them today. But understanding the patterns of disease using your local registry, the International Gaucher Registry, and then correlating that with genomic technologies to try and understand why one person's Gaucher is so different to the other. And of course, we want to pursue higher-end science with mouse models, stem cells, and being very ambitious and bold to develop the brain cell atlas of Gaucher disease. And this is what I'm going to be trying to describe today, hopefully in a light-hearted way, but not in a very overwhelming way. So thinking about the brain in Gaucher disease is extremely important because around the world, the neuronopathic variant, the alpha-4P, happens again and again because of its particular characteristic with the pseudogene, okay? And so, in fact, there are orders of magnitude larger number of patients than type 1. But we have had a very European-centric view of Gaucher disease because if you look in the world literature, 95% is of N370 Gaucher disease that arose about 10 millennia ago in Eastern Europe. And these are the two most commonly important mutations, but of course there are several hundred. And the pioneering work on this was actually done by Dr. Barringer and Dr. Gibbs at the NIH, who discovered these two mutations and published in the New England Journal of Medicine as well as PNAS. So this is important, but sadly there have been the neglected members of our Gaucher community. And so it is a major unmet need. It causes life-threatening disease as shown in this child. And look at him. after 14 years of enzyme replacement therapy. Clearly, the disease has been modified, and we are seeing a new phase of Gaucher disease. But it is very hard to predict the disease based on understanding of the mutations. And the pattern of neurological involvement is very diverse, from just slight squint and eye movement abnormality to very severe disease, as many of you know. And I would say that our understanding of how individual cells in the brain respond to Gaucher mutation is very rudimentary. And we don't have biomarkers. We don't want to diagnose neuronopathic Gaucher disease already when the eye movement disorder has taken place, or the child is having really quite advanced seizures and so on. We should be able to look at a blood test and predict that, much like we do with type 1, measuring lysoGL1. We need markers that go from the brain into the blood, and we can have a handle of where the patient is heading to. So the brain is a very complex organ. But the epicenter of Gaucher disease pathology, one might argue, is in this region. So that is the basal ganglia, which controls movement, thalamus, which controls, regulates sleep, consciousness, and alertness, hypothalamus, which controls body temperature, hunger, and sleep, amygdala controls memory, decision-making, and emotional response. When I lose it sometimes and get very excited in my household, my younger daughter tells me, "Dad, you are suffering from amygdala hijack," because the amygdala is really firing when you're worked up, okay? And the cerebellum. And this area, the brainstem in this region, appears to be the part of the brain that bears the brunt of the GBA mutation and the lipid fatty substances that build. When we look at the individual cells, it really is a most beautiful cooperation of cells. So there are neurons which fire impulses, help us to make circuits, memory, show emotion, everything. This organ is the seat of our emotion. Okay? And then the neurons are covered in myelin sheath, which are membranes that go round and round to insulate the neurons and facilitate nerve conduction so that the nerve impulse goes down at a lightning speed. Why is that important? Because the myelin is full of fatty substances. It's full of cholesterol and glucosylceramide and other sphingolipids, okay? And then there are astrocytes. So the brain, although it's such a small organ, has 50% of blood cholesterol, and it doesn't get cholesterol from the body. It is made inside the brain, and astrocytes are the master regulators of meeting the cholesterol needs of different types of cells, okay? And so these cells are cooperating and working in a beautiful orchestra to make us who we are. And I think we have only rudimentary understanding of where in the brain GB has the most impact, what regions, what cell types, and what is the sequence. So if you have a child, God forbid, with type 3 Gaucher disease, you want to think about what cells are impacted most, what biomarkers you can monitor, what will be the therapeutic approach. So the brain is very complicated. And so if you just look at the mouse brain, it has 70 million neurons, but all the other cells, the oligodendrocytes, microglia, astrocytes, outnumber neurons 10 to 1. So in fact, they used to be called cells just to glue microglia as neurons together, but actually they are the central places that maintain brain health. Okay? And so in order to understand the brain in Gaucher disease, what we've taken the approach is to develop mice with Gaucher disease that has deficiency of GBA in all cell types of the brain, and then we rescue GBA in selective cell populations. And we also have a model that's a healthy mouse in which we can selectively remove the GBA gene in the microglia to see what is the impact of that in brain function and brain health. So doing this, we were able to, using new technology called single-cell RNA sequencing and single-nucleus RNA sequencing, which allows us to take the brain, isolate nuclei, and then map individual types of neurons and individual types of microglia and so on. And so we used these mouse models, and what we learned was that the three main cellular targets of Gaucher GBA mutations are the neurons, but also astrocytes, microglia, and the dying neurons. And the microglia are, if you like, the valiant warriors of the damage that's going on, because the microglia are trying to mop up all this lipid that's generated to calm the brain down, which is very inflamed and is getting damaged. But there are other cells that come into the brain to amplify the effects, the immune cells, for example, and also the microglia, in their efforts to restore health, get converted into what's called damage-associated microglia, which paradoxically worsens brain injury. And we've been able to map out a range of disease pathways, lipid metabolism, not just the glucosylceramide, but also cholesterol metabolism and so on, as listed here. And so the microglia are one of the most important cells. You need to think about the microglia in your day-to-day life. And by the end of my talk, I hope I will convince you, because these microglia cells are tightly connected with the rest of your body, including even social enrichment, which we are doing here in the Gaucher Community Alliance, okay? So the microglia has a Dr. Jekyll, Mr. Hyde personality, okay? So Dr. Jekyll is a good guy. He's doing all the good things. It's helping your neurons to make connections, dealing with any harmful forces like buildup of glucosylceramide, viral infections, and so on, any kind of stress. And it allows the neurons to make synapses and connection to build the memory pathways, okay? But they have a Mr. Hyde character. And this Mr. Hyde character has been described in Alzheimer's disease, Parkinson's, multiple sclerosis, ALS. But it is now, we know, also present in Gaucher disease. And that conversion from Dr. Jekyll to Mr. Hyde is driven by the Gaucher lipids. And if you can fix that, you can restore the microglia to health, okay? And so there are ways to really map these cells based on these markers that are highlighted. But what we've learned is that the microglia convert into not just the two main types, the damage-associated microglia, but there are many different subtypes of microglia. And this one's shown here is lipid droplet-associated microglia. So lipid accumulation is a central feature of neurodegeneration of many different types. Okay? And so the challenge for us is to understand all the different stages of health, just like we do with bone disease. We want to think about bone pain, marrow infiltration, osteoporosis, psoriasis, right? And fractures. Same way in the brain, one should think in a stepwise manner of what are the stages in development of neurological disease. Okay? So what we want to really do is to understand what parts of the brain are affected. We want to understand what cell types are affected and what is the sequence. And I hope that if we have that understanding one day, we are going to be able to have a panel of biomarkers. So you go and have blood tests and it may give you a handle into your brain health in general. Okay? So this is a technology called spatial transcriptomics. Wild type stands for healthy mice, and GD stands for the Gaucher mice. And you can see at the front, the olfactory bulb is at the front, and then cerebellum is the back. But basically, you can resolve all of the structures of the brain, but more important regions of the brain are shown here. You know, the hypothalamus, thalamus, midbrain, medulla, and cerebellum. And then in the right-hand corner, and we have that kind of structure preserved in neuronopathic Gaucher disease. And in the right-hand column is this map, a beautiful universe that opens up when you isolate the nuclei and then sequence individual nuclei that corresponds to different regions, okay? So this is kind of a little overwhelming science, but we are going to meditate now. I'm going to show you a video. It is 4 minutes, but because of respect to Cindy and Viva, I'm going to stop it too, but I guarantee you, you're all going to say, please don't stop, okay? So I hope this works, okay? So the idea here is to try and explain this single cell sequencing technology, which is truly revolutionizing whole of medicine. Okay? What happened? It was working. I haven't dragged it. Thank you. Okay, so this is a video about fascinating birds called starlings. Okay, so you think about starlings flying all over the place. Then you have a registry, so you see patterns of disease, and you collect number of patients to understand how the disease works. But we now want to understand how individual cells behave in the setting of disease. And this dance of the starlings, if I can get it to work, is a very beautiful depiction of modern technology of single-cell RNA sequencing to show how the disease unfolds, how the protective cells kick in, and then repair takes place in a very dynamic way, rather than just taking one off at the end after seizures have occurred in type 2 Gaucher disease, because all the stages have been bypassed. But we should be able to follow each step because it will reveal unique opportunities for treatment. So it's not going to work. I got it to work. Okay, go back to the slide. So sorry about this. Please forgive me. I hope I'm not going to take everybody's time. I think this is something to do with the connection. It's a what? Yeah. I didn't get it ready for you. It's a sidebar. Could you try it? Those other two girls, when they get it. Okay, great. Okay. I'll stop at 1 minute because I wasted a lot of time. Can I have the
No, that's 1 hour. Yeah, yeah, you're good.
It's,
I don't know.
when you do this single-cell
Incompetence of
technology in the
the
lab, it
whatever.
opens a
Yeah.
whole
No,
new
you get it.
universe of,
No, you.
of what is happening. You right? And and this is, thank you so much. And that's almost like this. These different groups of starlings, you can think of it like a different populations of cells. And we can convert that into mechanisms of disease, understand what cells are involved in disease. But this is such beautiful music. So I'm just going to let you meditate for 1 minute, okay? Okay. This is on the internet, the dance of the stars. Keep on listening. I knew you were going to say this. But it's on the, you can, if you Google Dance of the Starlings, you'll be able to see it, okay? All right, so this is the technology, okay, of what's called spatial transcriptomics. Okay, this is where we take samples of the brain and put it on a slide, and there is a technique to look at different regions of the brain and how they are affected. Not only that, we can identify what cells are affected, what genes are driving the disease process, and then try and understand using this technology, ligand receptor interaction, how the cells are talking to each other. Okay? And so we did this, and we were able to identify the microglia, the astrocytes, oligodendrocytes, and neurons, different types of cells, but also looked at different functions in the brain, like the synapse formation, the connections between neurons, the lipid metabolism, and also the blood-brain barrier that you all know about. Okay? So here are the microglia. Okay? So the first thing to say is that the blue is normal, but when you start to see red and yellow and green, that means there is exuberant signal relating to each of these genes. GPNMB, which is a biomarker of Gaucher disease, much like chitotriosidase. There are other markers like cathepsin Z, this osteopontin and galectin, which are also markers of this bad Mr. Hyde microglia that I talked about. Okay? So even the astrocytes are abnormal because we find the astrocytes are very activated. And this is a marker of astrocyte that lights up when the astrocytes are under stress and cannot do their function. These are some of the other markers of astrocyte function. And so the normal physiological function of astrocyte in cholesterol metabolism, helping synapses to form, maintain the blood-brain barrier, is severely disrupted in neuronopathic Gaucher disease, okay? So now we look at the oligodendrocyte, this fascinating cell which wraps lipid membrane around the neurons, and you see there is a marked reduction in the staining for oligodendrocytes, that oligodendrocytes are severely injured in the setting of Gaucher disease. And so the normal processes of oligodendrocytes, like forming axon myelin and maintaining cholesterol and immune homeostasis, is severely disrupted. And there is also neuronal loss. So, of course, we all know that there is neuronal loss, so this is not a surprise, but there is a marked reduction of Purkinje cells, which are cells in the cerebellum. The neurons, believe it or not, are constantly renewing, particularly in children, and that differentiation is affected quite severely. Neurofilament is a marker of neuronal health that is decreased, and calcium signaling in the brain in NGD is severely disrupted. And so neurons are severely impaired, but there is a lot of neuronal stress affecting the synapse and the neuronal health. Okay? And so we tend to think that Gaucher disease is only about one enzyme. You have your enzyme treatment, you have your GBA mutation, and that's the only lipid. But the biggest surprise is that Gaucher disease has a widespread effect on lipid metabolism in the brain, one of which is reduced cholesterol production. And if you don't have enough healthy amounts of cholesterol in the brain, it is going to be affected. There's also an effect on fatty acids. Fatty acid hydroxylase is reduced. And this enzyme, which paradoxically increases the production of glucosylceramide. Okay? So what we can do is take this data and then ask the question, how do the cells talk to each other? Because if we understand that maybe there are ways we can intervene or we can follow patients better. And so this is all of the abnormal signals. And what were downregulated are shown here. And the ones that light up are GABAergic neurons, glutaminergic neurons, and also the cholesterol pathway is markedly downregulated. Claudin. Claudin is a marker of blood-brain barrier integrity. So in neuronopathic Gaucher disease, blood-brain barrier is leaky. It actually allows immune cells and other things to go through. And so this way, using this, we can develop interactions in different regions of the brain in the healthy mice and in the neuronopathic Gaucher disease. And when these interactions are thin, shown by thin lines, it shows that these interactions are severely impaired in disease. And these are some of the signaling pathways that are affecting lipid metabolism, synapse formation, as well as the blood-brain barrier. So what are the bad actors? What is driving some of the disease? And it is Dr. Jekyll, Dr. Hyde microglia, see? And so these are all upregulated: galectin, complement, granulin, SPP1, and so on, see? So then using this kind of pathway, you can see what are the main pathways that are driving the disease process. And the top one that comes up is apolipoprotein E, which is a cholesterol transporting pathway. And interestingly, variations in ApoE have been linked to Alzheimer's disease. But cholesterol transport is severely affected, and the osteopontin that I talked about. Okay. And this is the way we kind of are starting to map the brain cell atlas in Gaucher disease and understand the molecular underpinnings of this. And so the healthy brain is a wonderful orchestra of different types of cells talking to each other. If there is stress, cells like microglia come in and repair the damage and allow to promote brain health. In neuronopathic Gaucher disease, first of all, the blood-brain barrier is disrupted. It is allowing immune cells to go through, which is aggravating neuronal injury, but also these oligodendrocytes are severely affected, as are astrocytes, and the Mr. Hyde microglia, the damage-associated microglia. Okay. And so what's next? And what we are able to do is from human skin biopsy, we can generate iPSC cells, and grow microglia, neuron, and astrocytes. And I can also tell you that we are doing this for oligodendrocytes, and study these cells individually, okay? But also we can mix them up and study the interactions of the cell. So colloquially, one might say this is like a brain in a dish. But those kind of ambitious studies, I think, may be productive in helping us to advance. This is a Gaucher Community Alliance meeting. I mentioned right at the beginning that social enrichment that this meeting represents is very good for brain health. There was this incredible study published in mice. One set of mice just ate, drank, and slept. The other set of mice had toys to play. And the mice that had social enrichment had less microglia activation and inflammation. Think about that. So now we know that your gut health, health of your spleen, the liver, cardiovascular system can all affect microglia activation. So how you take care of your general health will affect your brain health. Very simple take-home message, okay? I couldn't do this without this small team of super dedicated individuals. So I wanted to just thank them.